So hi, my name is Jason Williams. I am an assistant director here at the Cold Spring Harbor Laboratory DNA Learning Center. Photo of the lab is behind me. And I want to welcome you to the May Meet a Scientist series. I'm just going to share my screen, just briefly introduce a little bit of information for everybody. You probably have come to the Learning Center's website where you can see more about this program. So here we are in the Meet a Scientist series. And today we're very privileged to have Dr. Hannah Meyer, who's going to give you an introduction to herself in just a moment on her topic about educating the immune system, how T cells distinguish friend from foe. And I also want to direct you back to the Learning Center website because you can visit, actually, I shouldn't have gone away so quickly, but back in our Meet a Scientist series, we have one more speaker who'll be joining us, I can already say it, next month. And that's Dr. Peter Koo, who's going to be talking about some artificial intelligence stuff. So you can already go ahead and register for the next talk about the same time in June, and that's the last one for the school year. Back on the Learning Center webpage, you'll also see that we're gearing up for the summer. So if you haven't had a chance, which many of us and students have not had a chance to do some hands-on science, you might consider joining us for the summer camps, and you can learn more about that information there. So with that said, I am going to turn it over to Dr. Meyer, and let me go ahead and replace the spotlight on her and welcome her to our series. And I'll turn it over to you to tell you a little bit about her life as a scientist and her work in research. Thank you so much for joining us, Hannah. It's all yours.
Thank you, Jason. And thank you for everybody that's joined the seminar today. I'm excited to tell you a little bit about what my research group does here at Cold Spring Harbor and how I actually got to do what I do now. And in order to tell you a little bit about this, I'm actually going to start off with where I'm from and how I moved from various places to get to Cold Spring Harbor. So you can probably tell by my accent that I'm not from around here. And in fact, I grew up in Germany, in a very rural area, very close to the French border, where I went to high school. And the high school system in Germany is quite different to the one in the US. But at the end, we have to choose 3 main subjects in which we will take A levels. It's similar to AP classes. And I couldn't quite decide what I wanted to do. So I knew I really liked natural sciences. So I did math and biology and English, but I tried to keep the other sciences as well. So I also had chemistry, but I couldn't take physics. And this sort of continued afterwards when I was trying to decide what I wanted to do for college. I knew I wanted to stay in science. I really liked biology. I thought maybe chemistry or pharmacy. So I couldn't quite decide what I wanted to do. And then I found a degree that has this fancy name, molecular biotechnology. And when I looked at the curriculum, I saw that basically it covered all the things I liked. There was a lot of math, a lot of biology, chemistry, physics. And some of the lectures were also going to be in English. So it all seemed like a really good choice. So I applied for this program and got into the bachelor's program in Heidelberg, Germany, where I did an undergraduate degree in molecular biotechnology. And towards the end of this, I wondered what I might want to do afterwards. And it soon became clear that if I wanted to stay in science, it would probably gear towards ultimately doing a PhD. But in order to do a PhD in Germany, you actually have to do a master's degree. You can't enter a PhD program with just an undergraduate. So I stayed in Heidelberg for another 2 years to do a master's in molecular biotechnology. And as part of my curriculum, I had to do a rotation in bioinformatics. And up till then, I had no experience with this. I was really not very interested in it. I really liked being at the bench, doing lots of experiments. So it was one of the things that I thought, oh, I want to get over with as quickly as possible. And indeed, like the first 2 weeks, I couldn't get anything done. I was pleased when I produced like 2 lines of code a day. But then I gave myself a chance. I was like, oh, this is something new. Maybe you should just enjoy it, learning something new. And then I really got the hang of it. And I thought it was super cool that we can actually study biology by using computational methods, developing new mathematical models to describe the data that we actually generate in the lab. So after this rotation, I actually ended up switching my major to bioinformatics. But most my training there was based on rotations and internships. So I never had a full, a good foundation for the bioinformatics based on courses. So I thought when I was applying for PhD programs that where I wanted to go should really be a place where I can delve in deep into the bioinformatics and immerse myself into a surrounding where all they do is bioinformatics and computational biology. So I applied at the European Bioinformatics Institute in Cambridge in the UK, and I got accepted in this program. And I spent 4 years there doing a PhD on quantitative genetics and trying to describe how our genes influence our heart function. And I really enjoyed it, and I learned a lot of methods. But I've realized that heart is really not, or studying the heart is not the biology that gets me very excited. But the one field of biology that really gets me very excited is immunology. So for my next steps, I really wanted to be able to combine my skills that I acquired in computational biology and statistics with the biological question that's close to my heart with immunology. So I applied at Cold Spring Harbor as a fellow, where you can start your own independent research right after graduate school. And I was lucky to be offered a position here. So about 2 years ago, I moved to Cold Spring Harbor, where I started my own lab, where we now study the immune system, specifically how it develops. And we use lots of computational methods for it, but we also actually do some wet lab experiments again. So I've come full circle from doing only bench work to doing only computational work to now combining these two, which I think is a big strength. Something else I wanted to highlight here is that neither of my parents actually have a college degree. The German education system, as I hinted before, is slightly different, and especially when my parents were in school, you didn't even have to have the highest high school degree to get a good job. Slightly different nowadays, but Germany has a big apprenticeship system where many people actually don't go to college. So for me, going to college was already a first in the family, getting a degree, and not even speaking about getting a PhD. So I'm the first academic in my family, and it's been a challenge, but it's also been very exciting, and it's been really, my parents were very supportive of it. And despite not having a good view of what's happening in academia, they're very excited about the science that we're doing. So if any of you are considering studying science or going to college and just not having family members that have done that either, don't be shy, try it. If that's your passion, I think you should go for it. Okay, so let's go over to some science. So I told you I've come from heart genetics to now studying the immune system. And I've also told you I've come from this maybe more standard view of what a scientist or about a biological scientist does, sitting in the lab with lab coats and gloves and looking at microscopy images and maybe having lots of tubes with, in this case, bacterial solutions in them, to switching over to this is what I now mainly do, where I have, as my collaborator would say, black screen, where we code and where we do the analysis. And instead of producing nice microscopy pictures, we often just produce graphs like this that tell us something about the data that we're analyzing. And we use these methods now and these computational programs to understand the immune system. And to explain to you what we look at in particular, I just want to give you a brief pointer to the particular part of the immune system that I'm interested in, which are the T cells, one part of the adaptive immune system. And with COVID, you've probably heard much more about the immune system last year than you would have had generally. So those are cells that can very specifically distinguish whether a cell is infected with, for instance, COVID, or whether it's a healthy cell. But how do these T cells actually do this? So I've got a little schematic here where we have different tissues, muscle, lung, and liver. And then I'm presenting here just a cell of each of these tissues. And each of these cells has little receptors sticking out. This receptor is called an MHC receptor. And this MHC receptor is a way of presenting what's happening inside the cell to the outside, because the T cell can only see the cell from the outside. So all the proteins that are being made inside a cell are presented on this MHC molecule. So cells we have different types of protein in the muscle and in the lung and the liver. So I just indicated this in colors here. So all of these cells are perfectly healthy. So now we have a T cell that comes along and tries to interact with this MHC receptor and a snippet of the protein from the lung cell. But because the cell is healthy, the T cell wanders off and nothing happens. In the case of an infected cell, here, for instance, an infection with a virus like COVID, the T cell actually can recognize this binding, the little protein snippet from the virus that's inside the cell. So just like any other protein inside the cell is presented on the MHC molecule. And if the T cell can recognize this peptide, it suddenly becomes very activated and can do its effector function, which in this case would be kill the cell that's infected, so not more cells will be infected. But you can already see the big dilemma here. How does the T cell, particular its receptor here, the T cell receptor that interacts with the MHC molecule and the protein snippet, how does that know that red here, the red protein, is actually means infection, whereas purple, pink, and brown proteins mean healthy? And this in particular is what we study in the lab. So where do these T cells learn this? So they undergo an education, and the education of the T cells, where they learn to distinguish healthy from infected or friend from foe, is in the thymus. So T cells are born in the bone marrow, and when they leave the bone marrow, they're naked, like they don't have T cell receptors yet. All they know is that they have to go to the thymus to get educated. So think of them as little school kids. They go to the thymus. The thymus is an organ that sits here, right atop the heart, and they spend a couple of weeks there. And once they leave, they now have T cell receptors. So you can see the little spiky bits coming out of here. Each T cell has the same, every receptor on a T cell has the same specificity, so we'll be able to react against the same protein, whereas T cell receptors from different T cells have different specificities. So here just indicated by colors. So by making many, many T cells with many, many specificities, we can cover the wide range of pathogens that could infect us. And then, as we've seen before, once these T cells actually find the pathogens they can interact with, they will activate and will have the effector function. So following with the analogy, we had our little school kids, they go to the thymus and then they sort of graduate once they leave the thymus. But then what happens in the thymus? So a more detailed scheme of it, in the thymus, these little T cells, you can see here in gray, migrate through and interact with different types of cells in the thymus, indicated here in gray with the green outlines. And these cells, particularly the ones with the green filling, are what I call the educators of the T cells. So these cells are the ones that can tell the T cell what is foreign and what is not foreign. And the T cell migrates. We're following one T cell here in purple, interacts first with these cells and then goes to the educators. And if all goes well, if it is successful in making T cell receptors, and if these T cell receptors are considered safe, so they're not going to attack healthy tissue, then they can leave. But how can these cells in the thymus, they're called thymic epithelial cells, how can they do this? So these cells basically serve as a type of library for the T cells to interact with. And you can imagine them representing this library, representing the entire body, representing all the proteins that the T cells need to learn so they can distinguish it. And I'm going to give you a small example of how this library selection works. But first I want to tell you like 3 aspects of what my lab does in particular. And then for one of those, I go into a bit more detail, which is related to the library. So we've seen before that the cells in the lung make different proteins than the cells in the liver, make different proteins than the cells in the muscle. You can imagine proteins in the muscle need to be able to contract, proteins in the liver need to be able to metabolize metabolites, drugs, or medicine. So if every tissue has their very specific proteins they need, how come the thymus can represent all of these tissues to educate the T cells? So basically, how can this one thymus tissue serve as the whole library of all the proteins that happen in the body? So we in the lab, translating this analogy to the lab, we're interested in how do the cells in the thymus, what is the mechanism that allows them to express genes and proteins that are usually only found in very specific tissues in the body. And then the other question is, how is this library organized? How do these cells know which genes they should express? Do all cells express all the genes? Do they express them in certain areas? Those are all questions that we don't know yet. And then we also don't understand quite yet how these educator cells or the library, how they interact with the students. How important or how many interactions are there before a T cell really knows it's safe to leave the thymus? When does a T cell realize that if I leave the thymus now, I will most likely only get activated if I find something that's truly dangerous to the body? And I'm going to give you an example what I mean with this library and how the organization might be important. So those are books that I have at home. It's a New York City guide, a novel, a nonfiction book, and a cookbook. So they're all quite different. And I've just taken, from each book, I've taken a single sentence. So imagine this is our small local library. And what would be the best strategy if we want to learn all the words that are in these books? Do we need to read all 4 of these books in detail to learn all the words that exist? This goes back to the T cells. Do they need to look at every single protein or at every single cell to learn all the proteins that they could encounter in the periphery? So I've taken a sentence from each of these books. Very simple sentences. In the summer, there's scores of free events around town. It's from the library book, from the tour guide. Then we've got, at this point, I should explain what had happened to me since that summer day, which is from the novel. We've got have a look at a simple puzzle from the nonfiction book. And then the cookbook says, this dish really reminds me of summer. So they're all very unrelated. They all talk about very different aspects. But if we look at this, we can easily see that in 3 out of these 4 sentences, we have the word summer. And if we keep on doing this, we see there are many more words that actually happen in many of these sentences. So off occurs twice, all of these sentences end in a period. This is a common word, me, a. So despite those being very, very different kinds of books, we can see that there's common words between these books. So the same is true in the body. While each tissue has very specific functions and needs specific proteins, like we discussed before, like proteins that can contract, for instance, a lot of these proteins, a lot of other proteins are actually shared. So in order to learn everything that happens in the cells, in the body, there is quite a bit of redundancy. And how can a tissue understand this redundancy and take advantage of this? And we don't quite know yet. And it's really hard to study in the lab, but we can study this with computational methods. And I'm going to show you a small simulation program that we're working on in my lab to help understand how this concept of a small library or a big library in overlap can apply to T-cell selection. So the selection process I've been talking about is actually called central tolerance induction. And think of it as in the most simple sense, think of it as a bag of cells where we have our educators, the epithelial cells, that express all the proteins that could be encountered in the body. And then we have our T cells or thymocytes, and they interact with the epithelial cells, these tags. And in these interactions, the T cell receptor and the MHC, the T cell receptor on the T cell and the MHC molecule on the epithelial cell, together with proteins on the MHC, try to interact. And when the T cell successfully interacts with one of these MHC molecules, that means it would recognize a healthy cell. It would recognize a protein that is expressed by your own body. And then this epithelial cell actually tells the T cell that it has to commit suicide for the greater good, because it's not safe for the T cell to leave the thymus, because it could destroy cells in the body, something that we call autoimmune diseases. So the T cell keeps on sampling cells from this bag of cells in order to figure out if it can react with something and if this reaction is harmful. And you can see that if I only pick out a single cell, the chance of having an interaction is much, much lower than if I pick out 1,000 cells. So the number of random interactions will be quite important for figuring out whether a T cell has potential harm of autoimmunity. And then also, all of these cells produce the proteins from the body, but we've seen that there's always like little parts of the protein represented on the MHC molecule. So not necessarily all the parts of the protein are always available to T cells. So that's another parameter that we can include in our model. And then we can use the vast literature that's already out there on T cell development for people that have worked in the lab and figured out how many of these epithelial cells do we have in the thymus. So these are numbers for mouse, for instance. So we have up to 1 million of these epithelial cells. We have up to 10 to the 8 developing T cells at any given time. And then people have estimated that there's between 100 to 1,000 interactions. And then we also know how many protein coding genes are expressed in the thymus. Overall, those are the number of protein coding genes that we find in the body. And then we know how many proteins a cell can express at any given time. So using this very simple model and these numbers that we can find from the literature, we can then run simulations, and we can just say, if we have this many cells in the thymus and we give these cells the property of randomly expressing proteins, and then we see how many interactions do we need to cover the entire repertoire of proteins or antigens that we find in the body. And you can see here, if the number of interactions is in the thousands, then however much antigen is available, so down here, only 20% of antigen would be available, whereas up here, it's 100%. it barely matters. We cover the entire repertoire. It would be safe for the T cell to leave. But we see if truly the number of interactions that are possible, and we don't know this yet, is only 500, for instance, then the antigen availability plays a huge role. And we can see that we only cover just over 85%. And if you can think about that, a single protein that's missed might cause an autoimmune disease, missing almost 14% could have a huge implication. And the last bit that I want to show you here is, here we're assuming that this protein presentation is random, but maybe somehow these cells have a system of how much is expressed by what cell. And in this case, we can see if there is a system where certain cells express the same type of antigens, then despite having fewer interactions, we can actually push this number much higher. So even if at fewer interactions, we can cover more than 90%. So using computational methods like this can be really helpful to generate hypotheses that we can then test in the lab. And I've mentioned it a few times, this is a very simple model. And what we're working at the moment in the lab is generating some data in the lab that we can then use to not randomly selecting cells, but following a T cell through their migration and see whether spatial components of the selection process are important. And for simplicity, we've only looked at T cells and epithelial cells now. There's actually quite a number of different cell types in the thymus. We also have B cells and dendritic cells that you might have heard of. These cells secrete chemicals to attract cells, and there's also different types of T cells. So we've established a simple model, and now we're building up on this model to get a more and more realistic representation of what is happening in the tissue, to use these simulations to understand the biology. And once we can understand these healthy processes, then we can also try and understand what happens in pathological processes when the immune system is not functioning properly, either because its T cells don't recognize pathogens or because it recognizes self tissue. And I have one last slide that I want to show you of the work that we're doing in the lab that actually contributes to our model. So I've just spoken about, does the spatial organization and the spatial migration make a difference for the selection process of T cells? And in order to study this, we've taken mouse thymus and chopped it in very, very small pieces, illustrated here. And then from each of these pieces, we extracted the epithelial cells and we measured all the genes that are expressed in the section of the thymus. And then we checked whether the genes that are expressed in each section, whether they differ from one another. And if they differ from one another, that tells us that the migration path can't be random, or if it is random, then certain parts of the proteins might actually be missed. And here I'm going to show you some results. So colors indicate gene expression. If it's dark, genes are lowly expressed. If it's lighter, genes are highly expressed. And this is as a control. Those are genes that every cell in the thymus needs, and we see they have moderate to high expression, and that expression is fairly evenly distributed. So that's great. Then we have some proteins that are specific to epithelial cells. And we can see they're also moderately expressed and also fairly even. So that's reassuring. But then there are these proteins that are specific to certain tissues, and I've just chosen two at random here. And you can see that those proteins actually have very different expression across different sections. We go from very low expressed to moderate expressed to lower expressed again. So there is really quite a pattern here, and we're currently refining this. We're making smaller sections. And then once we understand this better, we can feed it back into our model to inform the selection process and see what we get in terms of how many proteins are covered. And I hope this has shown you a bit how we can actually use computational methods to understand immunology and how we can combine it with experimental approaches to inform the model. And those are the people in my lab that have worked on this. And a big thanks to them, and a big thank you to you for listening to the presentation today.